material like has a natural stopping point, or I know it's getting heavy, so I know you need to like chew. You need to chew it up. That's when I do quizzes. They usually are about every three lectures or so. Yeah. And you always know the time before. I'm not going to show up and say you had a quiz when I didn't tell you the time before. But if you're not coming to class, that may be a big surprise to you. Okay. So first little thing is that I wanted to show you guys this little video because we talked about cilia and microvilli last time, right? So this is kind of a cool little video that actually shows you cilia in action, this is actually the respiratory epithelium. It's pretty cool, huh? This is the respiratory epithelium. This is an apical view of the respiratory epithelium, and all the cilia are waving this direction, right? And those are like particulate matter that you're breathing in, that they're actually moving them up, which is pretty cool. So you can see sort of the functionality of cilia by approaching that. Okay, so go ahead. Uh-huh. I actually don't know. I don't know how quickly they move. I can look that up and see if it actually is recorded on that video. That's a good question. I don't know the answer. Go ahead.
So, in reference to air pollution,
yeah.
That's really the problem is the ones that are small enough to actually get stuck in the alveoli?
Yes.
So are those the ones that are just big enough to...
They are big enough that the cilia doesn't catch them, right? So if you had a rake, right, and you're waving it through the air, it'll catch a leaf, right? But it won't catch a pollen molecule, right? So the ones that are problematic here in Utah, especially like right now, except it's snowing, thank goodness, are the particulate matter that is way smaller than the cilia that ends up going deep into your lungs. But we do have one other protection mechanism in the respiratory epithelium, which is its capacity to produce what? Mucus. And we'll talk about mucus. We haven't talked about it yet today, but we will. So that's one other little thing, because mucus is sticky and it's not as dependent on particulate size. So yeah. So let's talk that last tissue that we did last time. I think, didn't we do? No, we didn't do pseudostratified yet. Okay, hold on to that question. We'll talk about pseudostratified in just a minute. Okay, so the tissues that we did last time were all the simple tissues, right? Okay, so we did finish the simple tissues, right? I think we did. We did. Yeah, because we did the ciliated simple, and we contrasted it with the straight simple columnar epithelium. Okay, and we're going to practice this a little bit after we get through all of the all of the epithelial tissues. I'll start throwing some unfamiliar figures at you. Before we do that, first thing I want to do is see if you guys are getting confident at telling the difference between microvilli and cilia on a slide. And there's no way to do that by looking at a single slide or a single slide for each tissue. So what you're going to have to do, and I'll sort of get you confident with like how I'm going to quiz you and what you can expect. And in general, expect me not to use a slide that you have seen before, but a slide that is a good representation. So this slide right here. Okay, what I will do for you, and listen, this is important. There are all kinds of places on the slide that can be relatively distracting. I am not trying to confuse you. This class is hard enough to pass without confusing you. So if I pointed to a specific location, try to focus on that location because there are other areas that look worse. And then I asked you whether that was cilia or microvilli. You tell me what? That is cilia. Good. Okay, let's practice a couple more. How about what's at the pointer here, cilia or microvilli? Microvilli, exactly. How about what's at C here? Cilia, yeah. So you're getting good at it. How about, no, I don't like that one. How about this one, cilia or microvilli? Microvilli. Do you see that you can get pretty confident at it just by sort of repetition? So you have lots of resources. You have the PowerPoint that comes with lab. You have the slides that are in lab. You have these videos that I will capture for you. You also have 3 different histology resources that I put up for you on the links page, right? And those are adequate. Am I going to guarantee that the histology slide that I use comes from those sources? No, because I don't want you to memorize. I want you to get to the point that you're confident enough to figure it out from a good question. And so that's my job, right? Make it a good slide, a good representation of the tissue. And you can pretty much depend on me trying to do that for you and pointing to the best region of the tissue for you. Okay, so you understand? Oh, by the way, this is a scanning electron microscopy that shows you the difference between cilia and microvilli. Isn't that cool? I love that. I don't know. I want to frame it and stick it on my bathroom wall. I have all kinds of weird anatomical art around my house. My husband is very patient. He's like, really? Really? I'm like, yeah, we have to put it up there. It's all about reproduction. It's fertilization with the sperm and the egg. He's like, really? Living room? And I'm like, yeah, totally. It's the miracle of life. Okay. So those are all the simple tissues. You should be able to look at this figure and figure out very quickly which one's simple, right? They're all simple, which one's stratified, cuboidal, and columnar. This isn't really obvious which one's actually like cilia and microvilli, but I think that that's what they were meaning. That one was cilia and that one's microvilli. There are better ways to look at it. Okay, so now let's move on to the stratified tissues. So the general trend is that the thicker the tissue gets, the better it is at protection, right? But the worse it is at what? Transport, exactly. Any kind of transport, absorption, secretion, everything, right? So as we start to move into the thicker and thicker tissues, you will see the functionality start to change, right? And the locations start to change. So the stratified squamous epithelia are the two that we're going to do first. There are two different kinds of stratified squamous epithelia. And hopefully you'll get to the point that you can confidently tell the difference between them before we leave today. So this one out of your textbook is what we call moist or non-keratinized stratified squamous epithelium. This tissue is pretty thick. Do you see? Apical surface to basal surface. And now you can ask yourself the typical questions that you ask yourself, and the answers hopefully should be yes. Now, can you tell that it's an epithelium? Yeah. Can you tell that it's stratified? Yes. Yeah. So stratified, don't look at the layers so much as, for me, look at the layers of nuclei. Because every living cell will have a nucleus. It doesn't mean you'll catch it in every cell, right? But you'll catch it in some of them, right? So this is obviously living tissue all the way down here, right? And how do you name this tissue? Not all, these cells don't look squamous, right? But this tissue is named for the shape of the cells at the apical surface. So it is a stratified squamous epithelium. Does that make sense? Okay. Now it's kind of arbitrary, but somebody just decided, it's just so that you know it's not like just willy-nilly named, because these don't look the same as you move down. Now this tissue is of course going to be pretty protective, but not very good at transport, and that's not its real job. You're going to find this tissue that are subjected to a decent amount of abrasion, but have a source of moisture, so they're not really worried about water loss. So the places that you would find this tissue, classical places, are lining the mouth, right? You've got lots of layers. If you impale yourself on a Dorito, rapid rate of cell division, and you don't even usually bleed, right? Because there's a whole bunch of tissue before you get to blood vessels, right? Distal end of the large intestine, because when you poop, you pull off cells, especially if you ain't eaten enough fiber, right? Distal end, and lining the vagina, right? Those are the classic locations that you're going to find this tissue. That make sense? So this is either called moist or non-keratinized. I don't care which term you use. They're both really commonly used. Moist or non-keratinized. And some people hate that word. They're like, no, never gonna use that word. It is moist or non-keratinized. Okay, so that's moist. Now I want you to contrast it with this one, and then I'll show you pictures and see if you can figure out which one's which. Okay, so this one right here is called keratinized stratified squamous epithelium. I'm going to turn the lights down a little bit. Keratinized stratified squamous epithelium. Okay, so this one is basically like the other one, plus a big layer of dead cells on the surface that are full of a waterproofing protein called keratin. Okay, so it's keratinized. So it's basically like taking the other one and painting it with clear nail polish. So you're going to find this tissue in locations in which you have the possibility of abrasion and friction, but also not a tremendous source of water, right? Moisture source, okay? So this tissue you're going to find on the superficial surface of the skin, or what is that layer called? You may or may not know it yet. Epidermis. It's called the epidermis. But if you played Magic School Bus with me and said superficial surface of the skin, it's still true, right? Okay? If you hadn't learned the word epidermis yet. So how do you tell that this was keratinized versus not keratinized? Well, please note that you don't see any dark staining nuclei, right? Until you hit right here, okay? These are the dark staining nuclei. So the tissue is all dead up there, and it's living from here to here, which is interesting because remember I told you that this tissue is avascular, and this gets really thick. This is like the thickest epithelium in your body. But diffusion doesn't tend to be much of a problem because how far does diffusion actually have to go? It just has to go right there, right? Right? So it doesn't have to go all the way to the top of this tissue. It just has to go to the living cells right here. So this is keratinized stratified squamous epithelium, right? Does that make sense? So let's look at a few of them and see if you can tell moist or keratinized, okay, with a couple of different examples. Okay. Okay, this one. Moist. How can you tell? Nuclei all the way to the top. Does that make sense? How about this last one? How about this one? Keratinized. You see those dead cells from here to here, and then you've got, right? That makes sense? Let's see. I have one more. How about, this one's confusing. Look carefully before you answer. How about this one? That's moist, yes. Now, you can see that it looks like it might be dead, but then if you look, you've got nuclei all the way to the top, right? Right? Does that make sense? Okay. How about this one? Yeah, that's a pretty obvious moist one, right? So do you see that as long as I choose a decent slide, you can tell the difference very easily between the two of them, especially if you don't memorize. You logically work your way through it, right? Good? Okay, so that's moist versus keratinized, stratified squamous epithelium. And then let's look at stratified cuboidal. Stratified cuboidal is super duper easy because it freaking looks stratified and cuboidal every damn time. So this tissue right here, so can you tell it's an epithelium? Yes. Can you tell that it's stratified? It's got almost two distinct layers of nuclei, right? Okay, and although you can't see the boundaries of the cells, they are certainly suggesting that they are about as wide as they are tall, right? So it's a stratified cuboidal epithelium. This is a longitudinal section if we did the same tissue with a, where did I put it? Cross section, you can still tell, can't you? Yeah? So this is a stratified cuboidal epithelium right there. Is that good? Okay, so that's stratified cuboidal epithelium. You find this tissue lining the ducts, not quack quack, but the other kind of glands, like the duct that leads to and from your salivary gland or mammary glands or those kinds of things. That's where you find this tissue. Okay, all right. Next one is stratified columnar epithelium. This is a really rare tissue. You don't find it very many places in the body at all. Here's the figure that your textbook has of it, and then I'll show you another one. So just ask yourself, can you tell it's epithelium? Yes. Stratified, pretty distinct 2 layers of nuclei, and they're oval, which is helpful, and the cells are taller than they are wide, so it's columnar. Good. That's stratified columnar. This is another one of stratified columnar. Where are you? This one is also stratified columnar epithelium. Okay, now this is a relatively rare tissue. You're not going to find it in most places in the body. The classic location of this tissue is just in the distal 1/3 of the male urethra. The female urethra is only about 1.5 inches long because it just has to go from the bottom of the urinary bladder to the external urethral orifice. But the male urethra has to go from inside the bladder all the way to the tip of the penis, which is about 8 inches. I'm not saying penises are 8 inches long on average, because I'm not a fool. But from inside to outside is about 8 inches. And the distal 1/3 changes tissue type to this tissue, to the stratified columnar epithelium. When we get to the reproductive tract, you'll kind of understand why that is. It's got some special functionality. The male urethra has both reproductive and urinary functionality, so it has to have some extra cleansing and protection capacity. So that is where you find stratified columnar epithelium. You find it a couple other places as well, but that's a classic location. And then the last of the true stratified tissues is named differently. Because if there's a rule in biology, you know there's going to be an exception. So this one is called transitional epithelium. And transitional epithelium, let's start with the figure in your textbook so that you can move from that one down. I'll show you the other one. Okay, so transitional epithelium is a weird tissue, and you really only find this one primary location in the body. It is going to be a stretchable tissue, and you find it lining most of the urinary tract, lining the ureters that go from the kidneys to the urinary bladder, lining the urinary bladder, lining the entire female urethra, and the proximal 2/3 of the male urethra. Okay? And this tissue, if you were going to confuse it with any tissue, what would it be? Yeah, moist stratified squamous epithelium. That's what I would confuse it with. But please note, the apical surface has dome-shaped cells, right? And that's pretty dependable. Although, since this is a stretchy tissue, I'll tell you that this is what it looks like unstretched. What's it going to look like if you stretch it? Exactly like moist stratified squamous epithelium. I ain't going to ask you a stretched picture because that's mean, okay? But conceptually, you can understand that it would look pretty much like moist stratified squamous epithelium. But when it's not stretched, you can tell it's stratified. It's called transitional epithelium. I'll show you another picture of it. Where are you, transitional? This is another picture of transitional epithelium, right? Go ahead.
Does transitional epithelium have multiple different shapes like cuboidal?
No, that's part of the reason it's named weird, is because the cells don't really look squamous, cuboidal, or columnar. So they just skipped that whole part of naming it. Does that make sense?
Yeah.
Okay. But do you sort of see this convention? That obviously it's an epithelium, right? It's got a free surface, right? And obviously it's a stratified epithelium, but it doesn't fit into any of the other categories. And it's got dome-shaped cells at the apical surface. So that's how you can get used to that one. Is that good?
Yeah.
Okay. So I don't think that one's very hard after you get a little used. If you stretch it, I can't tell it from moist stratified squamous epithelium if you stretch it. So I never ask it that way. Okay, so that's transitional epithelium. And then the last of the epithelial tissues is pseudostratified. And nobody can really agree what order the words go in. The way that I learned it was ciliated pseudostratified columnar epithelium. I don't care which order you put those three words in because the textbooks aren't consistent about it, but I would be delighted if you got them all, right? Yeah, ciliated pseudostratified, ciliated. You definitely need the word epithelium at the end. Doesn't really make any difference what order the words are in. Let's talk about this one. It is a pseudostratified tissue, which means it appears stratified, but it is not. Okay, so let's look at how that can occur. So here's this tissue. What you have with this tissue is that it looks like the cells are piling on top of one another. But that is only because the cells are different sizes. So you've got like some little bitty cells, and then you've got some big ones that like flop over them. And so it ends up looking like you've got multiple layers, but you don't really have multiple layers. That makes sense? And then the cells that reach the apical surface are going to be ciliated. This tissue that you are learning, they're always ciliated. We do have pseudostratified tissue elsewhere in the body that you're not learning. It's not ciliated. So does that make sense to you guys? So it's pseudostratified because it looks stratified, but actually everything is attached to the basement membrane. Yeah? They're all attached to the basement membrane. They're just weird shapes so that they don't look like it. Hence pseudostratified. Okay. So this is your next ciliated tissue that you're going to learn. This one is primarily found lining the upper portions of the respiratory tract. Yeah? And so, as we talked about before, what is the function of the cilia in this tissue in this location? Yeah, it's basically getting particulates that you're breathing in, waving up so that they go out. And the cilia here, they don't wave willy-nilly. They wave like directionally, right? They're waving up. That makes sense? So that's the function here. That's the primary location that you're going to find this tissue. Something interesting about this tissue and about respiratory irritation and especially smoking. One of the things that smoking does over long periods of time, how long is not dependable. Otherwise, you would know how long you could smoke before you should stop. One of the things that smoking does over long periods of time is it paralyzes the cilia. And what does that do to your likelihood of having respiratory irritation and infection? Increases the likelihood. Over time, you can actually change the respiratory epithelium type, and it ceases being ciliated at all. And when it regenerates the next time, it will make non-ciliated cells that look very different. And then you've got a permanent change that increases your likelihood of respiratory irritation and infection for the rest of your life. And no, nobody knows exactly how long. It's based on your genetics and your exposure as well. So, everybody good with that? So that's ciliated pseudostratified columnar epithelium. So since that's our last epithelial tissue, let's just make sure that we know the surface features, the apical surface features. We've already done 2 of them, right? So apical surface features are anything special that's going on the apical surface. And we, of course, already compared and contrasted cilia and microvilli. Okay, but the other one that we haven't done is goblet cells. Goblet cells you have been seeing, but I haven't been talking about. Goblet cells have this shape where they look like they're skinny at the bottom and they're wide at the top, and they actually are open onto the apical surface. They're actually not skinny at the bottom. What they are is the nucleus is usually at the bottom. Where are you, picture? The nucleus is usually at the bottom, so it makes them look skinny. Do you see that the nucleus is sort of taking up a lot of the space down there? So just a second. And so what they are going to do is these cells, they don't tend to stain very well, so they look clear, like a goblet, right? And they make vacuoles filled of a protein called mucin, which when you mix it with water will produce mucus. Good? And then they dump it onto the apical surface. Go ahead.
So those are associated with just pseudostratified?
No, they're associated with 2 tissues that you learn. I'll show them to you in just a second, okay? All right, so can you recognize a goblet cell? Okay, so goblet cells are associated with a couple of tissues that you learn. One of them, of course, she was right, is pseudostratified ciliated columnar epithelium. Okay, so in this context, in this tissue, what is the mucus doing? What is the goblet cell doing? It's producing mucus for what purpose? Yeah, to catch irritants and all kinds of like aerosolized respiratory irritants and that kind of stuff, right? So these 2 are a team, the cilia and the goblet cells here. But you also find it, although not always, you also potentially find it in, where the heck is it? One other tissue, and that is this one, okay? So let's try to remember what this tissue is and where you find it, and then let's see if we can hazard a guess as to what the goblet cells are doing in this context. So what tissue is this? It's simple columnar epithelium, right? And give me a great location. Be more specific. Lining the small intestine, okay? And now figure out in that context what the mucus and the goblet cells are doing there. Because they're not doing the same thing as they're doing in the respiratory tract, right? Yeah, it's lubrication. They're doing lubrication. And mucus can be used for either or both. Doesn't make it, you don't really need any lubrication up here, right? Considering that you're bringing in primarily gases, right? So here it's actually lubricating. Does that make sense? Okay, so are those the only places that you are going to find goblet cells? No, but it's the only ones you're going to learn about. Are you going to see goblet cells dependably in every single slide? Absolutely not. So you can't use it as your only criterion. You can use it as a check mechanism. If I run into goblet cells, it could be one of 2 tissues. Does that make sense? So let's, to clarify this and make it so that you're not memorizing 13 different epithelial slides, every time you see cilia, it has to be one of 2 tissues. What are the 2 tissues? Ciliated pseudostratified columnar epithelium, or ciliated simple columnar epithelium. So if you see cilia, you've got 2 choices, not 13. Good? Okay. If you see goblet cells, you've got 2 choices, but they are not the same 2. What are they? Ciliated pseudostratified columnar epithelium and simple columnar epithelium. Yes, but not with cilia. Does that make sense? So do you see like how my brain sort of works? It's like when I look at a slide, I'm like figuring out not that, could be this, narrow it down, right? Then it's logic, not memorization, right? It's basically like a little dichotomous key in my head in which I'm like working through it to figure out what it could be. Microvilli. You only learned 1 tissue with microvilli. If you see microvilli and you're confident that it is microvilli, then the tissue is what? Simple columnar epithelium. Good? Okay, so let's practice just a little bit before we move on to the cell junctions. So I'm going to go ahead.
Is there ciliated cells in the intestines, though?
Are there ciliated cells in the intestines? Yeah? You don't learn any of them.
Okay.
Yeah. Well, I won't use any tissues that you don't learn.
I was trying to differentiate between the simple.
No, you won't learn any instance of ciliated cells other than the two that you just saw. That's it. Okay, so let's practice a little bit and see if you are confident with telling me a little bit about tissues. If you look at this particular location, what tissue is this? Yeah, this is ciliated simple columnar epithelium, right? Now, why did I point here? Because if I point over here, it can be misleading, right? So you have to trust me that I am actually trying to direct you to the area that is most indicative of the tissue, okay? How about this one? Moist or non-keratinized. Say all the words. All stratified squamous epithelium. Moist, stratified squamous epithelium. Okay, so what happens if you leave off one of the words? Yeah, the lab instructor can count the whole damn thing wrong if they want to. And usually I take off at least half if there's a word missing. Okay, how about this one? You can look anywhere, really. Ciliated, pseudostratified, columnar epithelium, right? See how smart you are? You're brilliant. You are brilliant, I tell you. Look right here. Non-ciliated or whatever, blank. Blank what? Simple columnar epithelium, good. Okay, this one. Simple cuboidal epithelium. All right, here's one. See if you guys can do this one. How about this one? All the words. Simple squamous epithelium. Yes, good. Feeling better about it? Are you feeling good about yourself? This one. Transitional epithelium. Good. Okay, so do you see that with exposure and practice, this actually gets pretty confident? It's not just random crap that you're seeing there. You're like, yes, yes, yes. And the first few times you miss it, and then by the time you've done it 15 times, you're like, I'm pretty damn confident I get this right almost every time. And if I choose a good representative slide, then we should be just fine, especially if you have practiced a lot. Good? Okay. Any questions? Last thing I'm going to tell you is how the cells are stuck together. Okay, so this concept is called lateral cell junctions. Okay, so what we have talked about, let's use a tiling of floor metaphor for a second. Okay, so what we've got when we have an epithelium on top of a connective tissue is that the connective tissue, right, and the superficial surface of the connective tissue, in order to stick the epithelium to it, like tiling a floor, you are going to have to put down adhesive, right? And the adhesive in an anatomical context is the what? What is it? It's the basement membrane, exactly. So, and then you're going to put the cells on top, but remember the cells are right next to one another, right? So what I haven't told you is that in addition to a continuous tile floor, there's also grout between them, right? And there's different kinds of grout depending on what you guys want to accomplish with it, okay? So what I want you guys to imagine is that this front row right here, you guys are all the cells in an epithelial tissue. And then I am a pathogen, and I would like to get through the epithelial tissue to infect the deeper cells. Everyone behind here are cells within a wet tissue. Yes, you are all within a connective tissue, okay? And what you guys are going to do is there are multiple ways that you could potentially protect. Let's talk about the first way. The first way is a type of grout that you use to stick the cells together, and it's right here. Do you see? It's like a three-dimensional zipper, right? So what's going to happen is these two are going to zip themselves up with a three-dimensional waterproof zipper, which will force me, if I want to get through the deeper tissues, I can't go between the cells, I have to go through the cells, right? A lot more control when you have to go through the cells than between the cells, right? So this type of cell junction, or grout, right there, has to go all the way through, right? Otherwise you'll just go around it. And this is called a tight junction. Tight junction? Is that okay? Okay. So a tight junction is basically like playing Red Rover. Will you guys hold hands for just a second? Ready? Do it. Okay. All right. I want to get through, but no, I'm a pathogen, and so they are saying, back off. Okay, so that's what a tight junction does, only does it three-dimensionally. Where are tight junctions useful? Where you have to have a tremendous amount of control over what gets into the deeper tissue, like the brain. The capillaries of the brain are made of simple squamous epithelium, really, really thin. But the cells are all zipped together with the tightest of tight junctions that we call the blood-brain barrier. And why? Because you take in all kinds of weird crap into your bloodstream, right? And you want to be able to protect your amitotic nervous tissue, yes, from this going through. So in order to get into the brain, if I were something really important, you would know who I was. Hi, I'm glucose. Can you let me through? And you're like, cool, I love glucose. Go ahead and go in, right? Hi, I am a trypanosome who wants to infect your brain tissue, and you guys just both flip me off, right? And you're like, out, out, out with you. Does that make sense? So that's a tight junction, okay? Tight junctions are about, if you associate words with them, associate impermeability, okay? Sometimes they're so tight they're even impermeable to water. Water can't get through, okay? So, but they are not strong, because if somebody yanks these two from the side, they would pop open, right? The zipper would pop open. So they're about impermeability when you're trying to traverse this time, but they're not strong this way. The next type of junction is stronger, but it's not three-dimensional, so you could go around it. So it's like a spot weld, okay? So what happens with this one, it's called a desmosome. It's about strength and resistance to stretching and twisting, but instead of a three-dimensional zipper, what happens is she would reach in and hold on to your cytoskeleton, right? And you would reach in and hold on to her cytoskeleton. And we would have a really strong spot weld, but a good person at Red Rover could go over it or under it, right? But you can't pull it apart, right? So this is about strength. Desmosomes are about strength. Where do you see those? In any tissue that is subjected to a lot of stretching and twisting, but you need the cells to hold together, you can use desmosomes. So your skin, for instance, has desmosomes in the lower layers, right? And then even though your heart isn't an epithelial tissue, those cells need to hang together even though they're going to fill and empty, fill and empty, fill and empty. So the cardiac muscle cells have desmosomes between them. And then the other location that makes it make sense is the cervix. So the cervix, you got like a nostril size hole, right, that you need to stick a freaking grapefruit through and ever be able to use it again. You better put some desmosomes in there, right? So there's a whole bunch of desmosomes in the wall of the cervix. Does that make sense? Okay, and then one last thing, and this one is this one. These are called gap junctions. Your textbook, your phys textbook calls them connexons, but I don't know, I've never used that term. They just came up with it recently. So these are called gap junctions. Here's what happens with gap junctions. They're not about strength, they're not about impermeability, they're about communication between the cells. And so what happens is, remember learning about channel proteins in 1610? So you just take your channel protein and put it together with her channel protein. So it's almost instantaneous communication between the cells. So if you guys are all in a row, right, and he gets a signal and he's connected via gap junctions, then you'll get it next and then you'll get it next. This is used, for instance, lining the respiratory tract, because if I start from the bottom and give him a signal to wave, wave, and then wave, 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 wave. So, but this is how you would actually be doing it. If I was starting from the bottom, you would be waving that direction, yes? And then you all would be waving, like in a coordinated motion, because you would all get the signal one after the other. Does that make sense? So, what if I needed both strength and communication? What would I do?
Use both?
Yeah, use both, exactly. If I needed impermeability and strength, I would use another two, right? So there's no rule that says you can't use more than one type of connection between the cells. And for instance, with cardiac muscle, I don't know if you guys know this, but cardiac muscle contracts in a wave-like contraction. So it has gap junctions for wave-like communication, and it has desmosomes for strength to hold them together. So you'll run into this throughout the semester, cells that have different types of connections between them. So these are called, by the way, lateral, because it's to the sides of the cells, cell junctions, right? How do they stick together? And I think of them as grout, but don't put grout on the test, because that's not the answer. Lateral cell junctions is the answer. Good? Okay. So Study tip. So here is what a poor student does for histology, and I don't want you to be one of those. They look at a histology image, and this is as far as they get. They go, oh, okay, keratinized stratified squamous epithelium. There's way more questions you could ask about a slide than that, okay? So what I want you to do in order to be confident and do well in both lecture and lab is for every slide, or 10 different iterations of the same tissue, be able to answer all of these questions before you move on. And so what kinds of questions are fair to ask about this? So what general type of tissue is it? Which means, don't forget the easy questions. There's only 4 general types of tissue, right? Epithelium, what are the other 3? Muscle, connective, and nervous, right? So don't miss the easy questions, right? What general type of tissue is it? It's obviously an epithelial tissue or an epithelium. Okay, next question is the one that everybody expects, which is what's the name of the tissue? Keratinized stratified squamous epithelium or ciliated simple columnar epithelium. That's as far as most people ever get, but there are a lot more fair questions that you could ask. For instance, any apical surface feature, what is the structure that you are seeing here? Cilia. Cilia, microvilli, goblet cells, all fair questions. Okay? And then what else? Oh, don't forget you also know what this is called, don't you? That's called the lumen, exactly. And then modified structures we did, surfaces, okay? So if we look at this tissue, what is this surface called? And what's this surface called? Basal. And then if I had a good one, the structure underneath there would be called basement membrane, right? So that's a fair question too. Here is what people do the worst at, and I don't want you to be one of those people. Specific body locations, okay? Specific body locations, you have to play Magic School Bus, you have to describe it in a way such that if I were blind, I would be able to understand what you were talking about. So with any tissue, for instance, with this tissue, give me a true statement about a specific body location for this tissue. Raise your hand. Lining of the oral cavity, good. Lining of the vagina. Would you get it correct if you just said mouth? No, that is made of more than one tissue, and that may seem nitpicky, but it ain't, because immediately we will learn that deep to that there's going to be a certain type of connective tissue, and then there's going to be a certain type of muscle tissue. So mouth is not specific enough for anatomy. So let's do just a couple more. Let's talk about this one, body location. It's pseudostratified, yes. Body location. Lining of the upper respiratory tract too. And I probably wouldn't even count it wrong if you didn't put upper because you haven't learned what's in the lower yet. But do you hear the important lining word that is there? Okay, so with, how about this one? Body location. Skin would not be correct. Superficial surface of the skin or epidermis would be correct. So do you hear? Okay, so you just have to be specific enough that there aren't any other true answers for the thing that you said, right? Is that good? So this is just another confirmation of if there is a more specific answer, the general answer is wrong. Go ahead.
I have a super silly question.
Okay.
If you were to show us a slide of this on a test, would you want us to label both layers, like the keratinized stratified and the living stratified?
Nah. Yeah, I've never really done that. It's interesting conceptually, but I don't really, it's actually not called different things. They're just trying to show you which ones are dead and alive. Okay, everybody good? You feel a little bit more confident about it? Okay, good. All right, so last thing about epithelial tissue is to talk about the tissue that's an exception to the epithelial rule. Remember, I've been talking pretty much about surface epithelium, right? Inner and outer surfaces. But I also told you that epithelial tissue made up glands. Yeah? Now, glandular epithelium is really hard to identify. It just looks like big blobs of cells that are really close to one another. So I generally do not make you identify glandular epithelium from slides, but you do have to conceptually understand that glands are made of epithelium. And then the first thing is, what is a gland? A gland is one or more cell that produces and secretes a product, which means we've already met a gland, right? What is the gland that we already met? A goblet cell is, by that definition, a gland. It's just a unicellular gland. So, but let's talk about the two big categories of glands. The two big categories of glands are endocrine glands and exocrine glands. and it has to do with where they dump their products, okay? An exocrine gland dumps its products onto a surface. It doesn't have to be an external surface, though. Like it can dump it into the digestive tract, for instance, right? Or the respiratory tract. Or the serous membrane dumps it into the peritoneal cavity. Does that make sense? Okay, so that's an exocrine gland. An endocrine gland dumps it into the bloodstream. Does that make sense? Okay, so the tissues look a little bit different, but with that concept in mind, exocrine onto a surface, endocrine into the bloodstream. I'm going to name you some glands that you have heard of before, and you categorize them as endocrine or exocrine, okay? Salivary gland. Exocrine. Pituitary gland. Mammary glands. Exocrine. Sweat glands. Exocrine. Thyroid gland. Endocrine. Do you see? That's how you categorize those. Okay. So you won't have to know them by histology, but why is it important that they fit into epithelium? One of the important reasons that you need to know that they fit in with epithelium is that remember that epithelium has a very fast rate of cell division and regeneration, which of course is clinically significant for likelihood of cancers, right? And so if we go back and revisit most of the common forms of cancer, they originate in epithelial tissues. They either originate in surface epithelium, like skin cancer, and like lung cancer, which originates in the epithelium lining the lungs, or they originate in glandular tissue, like thyroid cancer, and like pituitary cancer, and pancreatic cancer. Does that make sense? So it is clinically significant and relevant that those tissues go together, even if you don't have to memorize the slides. The slides are damn near impossible for undergraduate anatomy students to like get right. So I don't really make you memorize the slides, but I will tell you with exocrine glands, it's not that hard to recognize just from a drawing, not from a slide, that they are epithelial tissue. So let's look at them. Okay, so, sorry, not that one, this one. So let me show you two pictures of these guys. Okay, so just look at it globally for just a second. And these are exocrine glands. One of the things that's true about exocrine glands is they generally have ducts, not quack quack, but the other kind. Usually the ducts are lined with what kind of tissue? Epithelial tissue. The whole thing is made of epithelial tissue. Do you remember what kind of tissue you generally find forming the ducts? Stratified cuboidal epithelium. So a lot of times they have stratified cuboidal epithelium here, right? But this is so convoluted that it really doesn't fit into the categories that you can see really well, especially as it gets to these big blobs of it. But if you look at it, can't you actually figure out that that looks like an epithelial tissue, right? It's just a highly convoluted epithelial tissue. But this is only true of exocrine glands, because exocrine glands dump via a duct, which opens at a pore or something that looks like a pore onto a surface, right? And so they'll look like this. You don't have to know these individually at all, okay? But you do have to be able to look at them and say, oh, epithelial tissue exocrine, okay? That's all you have to do as far as that. And this one is just at closer up. You can see the duct. Okay, but endocrine don't look like that because they're nowhere near surfaces, right? Endocrine are just big blobs of cells that just happen to have a bunch of blood vessels in them, so whatever they secrete gets picked up by the bloodstream. So they don't really look like this, and they don't have a duct. Does that make sense? Okay, so if you had any exocrine gland, a goblet cell is the simplest exocrine gland, and that one doesn't have a duct because it doesn't need it. But when you're multicellular, right, you're going to lead to the surface via duct, that when you looked at it from a superficial aspect, it would look like a hole or a pore, right? Whether it's lining your digestive tract, or it's in your respiratory tract, or it's the salivary glands dumping into your mouth, or that little hole right here that's your lacrimal gland dumping into your eyeball, that you are looking at the top of the duct right there. Does that make sense? Okay, so that's endocrine versus exocrine. I am not holding you responsible for the types of exocrine glands, just that they are exocrine and epithelial tissue. Does that make sense? Okay, so tons of examples of endocrine glands. Okay, so what we are going to do next time, the other class is a little behind you, so I'll just give you an intro of what we're going to do with connective tissue. and then we won't get into the specific tissues until next time. Connective tissue is the hardest one. I wouldn't miss on Tuesday because I can help clarify for you and make you understand like why the hell blood and bone are both connective tissues. It is the most common tissue in the body. It's also the most diverse, and unfortunately, there is no consistent naming convention. There isn't, right? It's not blank, 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 right? It is named whatever the hell it's named. So the best that I can do for you is tell you the name and help you understand what that name refers to, right? But it's no consistent naming convention. One of the things that we will see is that the cells, different from epithelial tissues, you're going to have a cell and you're going to have a lot of space between it and the next cell. And the stuff between it is going to give the tissue a lot of its characteristics, right? It's going to make it squishy or brittle or whatever it makes it. That makes sense? So we'll start with that next time. And you guys can go. No quiz next time. No, not a quiz every time, just quiz like every third time. Or something.
Clarify.
Okay, hold on, let me save this before I forget to do it.